chapter 15 is dealing with the genetic code and translation. So let's get started. So this diagram is of the spleen, and the spleen is an organ found in the upper abdomen, and it plays a very crucial or critical role in defense against infection. Now, what does this have to do with what we're talking about? Specifically, there is a rare disorder known as isolated congenital asplenia, or ICA, and it is an inherited autosomal dominant trait. And specifically, it contributes to a disorder found in children that are born without a spleen. Now, this is associated with translation in that studies were done, and they found that there's a gene encoding ribosomal protein SA, or RPSA, and this protein is one of 33 proteins that make up the small subunit of the ribosome. Furthermore, when researchers began to study this more in depth, they found that there are several different types of mutations in the RPSA associated with ICA. Some cause premature stop codons, halting of translation before an actual functional protein can be produced, or even a frame shift mutation. We'll talk about more of these type of mutations in chapter 18. Okay, but ultimately, these different changes in the protein or in the DNA can ultimately result in changing the mRNA sequence, which will then alter the protein or the amino acid that's added into the protein. So understanding how proteins are made is essential to human health, as just one example. So there is relevance to what we are going to discuss today. All right, so you may have heard the concept of one gene, one enzyme hypothesis. However, we understand that this concept was initially proposed with limited information, right? And there's flaws to this one gene, one enzyme hypothesis because it assumes that all genes encode enzymes, right? Well, genes function by encoding enzymes, and each gene encodes a separate enzyme. However, after as science has progressed and we've done more research, we now understand that not all genes encode enzymes. There are some genes that encode proteins that are not enzymatic in function. So to be more specific and more accurate, the hypothesis has been updated to be one gene, one polypeptide. So to help with this initial understanding of the one gene, one enzyme hypothesis was a hallmark study by Beadle and Tatum. I would strongly suggest that you write this down, that you learn this concept because it is very important, and it is a hallmark study in genetics, and not just genetics, but even chemistry, biochemistry. So Beadle and Tatum used fungus to look at the complex life cycle of this yeast or bread mold to really understand what is the association between genes and proteins. So to do that, there was a study that Beadle and Tatum developed. To start the experiment, the question is, how can an auxotrophic mutation be isolated and identified? Okay, so in order to study this, they pretty much developed a method for isolating these auxotrophic mutants in this bread mold or a Neurospora. So how it works is they used a culture of Neurospora that was irradiated to induce mutation. So they exposed it to X-rays, which caused genetic mutations. These individual spores were then transferred to individual tubes that were containing complete media. We talked about what the different types of media are earlier in the semester, so this should be a review for you, okay? Those spores were then cultured and then transferred to tubes containing minimal media, okay? And the fungi with the auxotrophic mutations did not grow on minimal media, as you see here. Growth is indicative of the brown-like substance you see here, okay? However, when they grew fungi that had other fungi that had minimal media, there were some that did grow. So their results and what they found was that the Neurospora grew only when it was supplemented, the medium, with arginine. And this indicated that the mutant was defective in the synthesis of arginine, okay? Now, you'll see here there are different types of amino acids, but only when arginine was added, did they actually see growth? They did not see growth in any of these others. So they identified that the mutation could be identified by spores' ability to grow in minimal media when they were supplemented with a substance that they could not synthesize. So a way to kind of explain this a little bit, the plus sign indicates growth, a minus sign indicates no growth. So here you have orthanine, citrulline, and arginine. Okay, these are different, and these groups are indicative of different mutant strains. So in group 1, in each of these cases, there was growth in the tubes. However, in group 2, there was no growth when orthanine was present, but when citrulline and arginine were added, there was growth. In group 3, there was no growth for orthanine or citrulline, only when arginine was added. So this is a way to understand the growth of these auxotrophic mutants on minimal media when they're given various supplements. All right, so in order to develop a method to understand the relationship between genes and enzymes using Neurospora, they developed an experiment. And the question for this experiment is, what do the effects of genetic mutations on a biochemical pathway tell us about the gene to protein relationship? So here you have wild type, which is considered normal, and then you have 3 different mutant groups. Okay, group 1 can only grow on minimal media when it's supplemented with orthanine, citrulline, or arginine. Okay, group 2 can only grow on media when supplemented with either arginine or citrulline, but not orthanine. And then group 3 can only grow on media when supplemented with arginine. Okay, so here you have the spores that are the auxotrophic spores that are placed on these minimal mediums, either with nothing or with supplements. And you'll see here that the wild type, when it's just minimal media, or with supplements, you have growth either way because they're normal. So they have all of the enzymes and the components needed to substantiate their growth. However, in group 1, because they have the mutation that blocks the initial synthesis of orthanine, you don't get any growth on the minimal media with no supplement. But if it's supplemented with any of these 3, you get growth. For group 2 mutants, you don't get growth on the minimal media or even when the orthanine is added, because in this case here, there is a mutation that blocks that prior step, so you don't have the synthesis of it. So here you only get of citrulline, so you only get growth when citrulline or arginine is added. And then of course in group 3, the mutants only grow when it's supplemented with arginine because the prior steps are missing. Now, the question is, what are these steps? These steps that I'm referring to are the enzymes which act upon the precursors to produce the product. So for an example here, in the group 1 mutant here, there is a mutation in gene A which codes for enzyme A. And enzyme A acts on the precursor compound, the components in the minimal media that convert it to orthanine. But because enzyme A is mutated, you do not get growth. The only time you get growth is when they add in or supplement the media with orthanine. Okay, so ultimately what is happening here is showing the relationship between the genes and the enzymes. When the gene is mutated, the enzyme is not produced, therefore blocking the biochemical pathway and blocking or inhibiting growth of the Neurospora. So this was a hallmark study in that it really helped contribute to the understanding of the relationship between genes and enzymes. So what is the structure and function of proteins? Remember, not all proteins are enzymatic. There are some proteins that are structural based. They don't have enzymatic function, but they do have a purpose that is not enzymatic. In order for us to understand that, we have to go back to the foundation in understanding that proteins are polymers that are made up of amino acids. And these amino acids are linked together by what we call peptide bonds. The linear sequence of amino acids is considered the primary structure of a protein. And that primary structure can then fold to create secondary and tertiary structures. When you have multiple polypeptides, 2 or more, they can interact with each other to form what we call quaternary structures. So let's look at some examples. So you have in a firefly, and the light is produced from the firefly by producing a light producing reaction, luciferin and ATP, which is cleaved, thereby producing by the enzyme luciferase, and that is how you get this light source, or this light that is produced. Okay, the key here is the luciferase enzyme. B, you show it shows the protein fibrin, which is the major structural component that produces spider webs. Or C, you have here in this diagram here, or picture, castor beans, which contain a highly toxic protein called ricin. Okay, so each of these scenarios show you different types of proteins and their role. But even if we go beyond this example, you can talk about collagen, we can talk about keratin, which are essential in hair production, skin elasticity, even enzymes that help break down your food that you consume. Excuse me, hemoglobin, which is a quaternary protein that helps with the carrying of oxygen and iron throughout the body. So there are many functions to protein production. Okay, so in order to understand the protein composition, we have to go back to amino acids, because amino acids make up a protein, right? The combination or the linking of multiple amino acids contribute to this polymer that we denote as a protein. There is a common structure that is found in all amino acids that we're going to cover, but I want to point out these different amino acids, okay, and they're grouped based off of their characteristics. So there are amino acids that are non-polar, aliphatic. There are amino acids that are aromatic, meaning they have this ring-like structure. And these tend to be more bulky. Okay, so you also consider these aromatic amino acids as your more bulky amino acids. You have your positively charged amino acids, your negatively charged amino acids, and your polar uncharged amino acids. So these 20 amino acids can be classified based off of their characteristics. And specifically, you'll notice that they are in pink. The black portion, okay, of the amino acids are the same in every amino acid, but it's the R group, okay, the radical group or the side chain, that make each amino acid unique and give it its own characteristic that actually helps to provide function to proteins. Okay, so I want you to think about this as you're learning about these amino acids. This is how you get the chemical nature of proteins, and it is through the reactions of the side chains of amino acids that work together to form to carry out functions of these proteins, whether they're chemically attracting one another, whether they are donating or receiving, causing other types of reactions to occur. It is because of these R groups or side chains. So what is the basic structure of amino acids? It is right here. You have the alpha carbon. Okay, and off the alpha carbon, you have the amino group. And then to the right, you have the carboxyl group. And then off of the alpha carbon, you have a hydrogen molecule. And then on the other, you have a R group. Let me go back so that actually you can see it here. Okay, so here's your alpha carbon. To the left, you have the amino group. To the right, you have the carboxyl group. And keep in mind, this carboxyl group may be written differently depending on if it is bound to something else, or if it's attached to something, i.e. this R group will cause this to, the chemical bonding and how you view this to change. It can be written as COOH, COO minus, or C double bond O. And then you also have the hydrogen. Excuse me. So here you have 2 amino acids that are held together. And they're held together by a peptide bond. So I want you to know a couple things in understanding how a peptide bond is formed. You have the oxygen from the carboxyl end of one amino acid and the hydrogen molecules from the amino group of another amino acid that come together through a dehydration reaction to form this peptide bond between the carbon and the nitrogen here. Okay, so from that dehydration reaction, you have the release or the formation of water, and this is how a peptide bond is formed. Okay, so an amino acids are held together by peptide bonds, and it is specifically between the oxygen from the carboxyl end of one amino acid and the amino group, the hydrogens from the amino group of another amino acid form through a dehydration reaction, form a peptide bond. So as I stated earlier, there are levels, or there's a structural hierarchy to a protein. You have the primary structure of a protein, which is literally the linear sequence of amino acids that are held together. The secondary structure of a protein is where you have the hydrogen molecules, or the backbone of the amino acids that actually form together with they form alpha helix and beta pleated sheets to form the secondary structure, but it's between the hydrogen molecules of the core structure of the amino acid. So the R groups are not involved in the secondary structure of a protein forming. And then the tertiary structure is when it starts to take on a three-dimensional shape, and this is where the R groups actually begin to interact with one another, causing the folding and bending of this protein, giving it this three-dimensional shape. And oftentimes in tertiary structure proteins, you also have these domains or pockets where other molecules can bind to, like heme or zinc. And then you have here the quaternary structure, and this is where you have multiple polypeptides that come together, and they form a quaternary structure. A great example of this would be your hemoglobin. So let's go into now the genetic code, okay? Because the genetic code helps us to understand the link between mRNA and protein. How do we go from mRNA, which is RNA made up of bases, adenine, guanine, cytosine, and uracil, to now amino acids? So we have to understand how the genetic code was broken. What does it mean when we talk about, or you hear, degeneracy of the genetic code? Reading frames, termination codons, and the universality of the code. So one, breaking the genetic code, have to think about homopolymers, polymers that are the same. You also have random copolymers and ribosome-bound tRNAs. To understand that more, expound on that, there was a study done by Nirenberg, and they developed a method for identifying the amino acid that was specifically tied to what we call homopolymers. Okay, so that would be an example of homopolymers would be all three bases that are all U's, or three bases that are all C's or A's. So understanding when you have three bases that are the same, what amino acid do they correspond to? In that case, the question for the experiment was, what amino acids are specified by codons composed of only one base? Okay, and so what they ultimately did was they added the various homopolymers to different tubes and incubated it at 37 degrees, allow for translation to take place, and then try to use a filter to check for radioactivity. They repeated this 20 different times with different tubes for different labeled amino acids, and they were able to look for radioactivity. And that radioactivity, which is synonymous with that amino acid, suggests that there was a newly formed or newly synthesized protein for that specific homopolymer. Okay, in this case, UUU was associated with phenylalanine. Okay, they also found in other experiments that AAA encodes for lysine and CCC for proline. And that's just a summary of this experiment, but I strongly implore you to go back in the textbook to read more about this, and it's figure 15.8. To follow up with their experiment and tying it back to the ribosome-bound tRNAs, that's the other part of breaking the code, Nirenberg asked the question in the experiment, with the use of these tRNAs, what other matches between codons and amino acids could be determined? So they use very short mRNAs with known codons. And then they added to that mixture ribosomes and tRNAs attached to amino acids. The ribosomes were then allowed to bind to the mRNA and tRNAs in a specified manner. The mixture was then passed through a filter, and they were able to then look at the paired ribosomes that were bound to mRNA. And what they found was when an mRNA with GUU was added, the tRNAs on the filter were bound to valine. Therefore, they were able to conclude that GUU specifies valine. And they did this several times for different other codons to determine what we now know as the genetic code. And so this is how these two experiments were able to enable them to understand the genetic code, and hereby forming what we know as the genetic table or the genetic code. A codon is a triplet RNA code, so 3 bases. Okay, in the table there are 64 possible codons. 3 of the 64 code for stop codons. That's UAA, UAG, and UGA. And then you have 61 sense codons. This means that they code for amino acids. Now, before I go into and showing you the genetic code, I want to point out a couple things about the degeneracy of the genetic code, because this is involved in understanding the genetic code. When you hear the term or the concept of the degeneracy of the genetic code, it's suggesting or referring to the ability of amino acids to be coded by more than one codon. Okay, there's more than one way to get the same amino acid. Synonymous codons are codons that specify the same amino acid. So you have codons, there's more than one way to get the same amino acid. And then you have isoaccepting tRNAs. And this is where you have different tRNAs that accept the same amino acid but have a different anticodon. And that also goes back to the shape and the structure of the tRNA. So with the codons, you have sense codons, okay? And these are codons that encode an amino acid. Then you have initiation codon. This is AUG, and it codes for the amino acid methionine. That is considered the universal codon, start codon. And then you have the termination codons, as I previously stated, UAA, UAG, and UGA. These are termination codons. They are not associated with any amino acid. They are specifically for termination of translation, and they are affiliated with release factors. Okay, and you'll see them denoted in your book as RF 1, 2, or 3. And then we also have the wobble hypothesis, and the wobble hypothesis is referring to the tRNA in that third position that doesn't always fit properly into the ribosomes when it is being read. So let's look at the genetic code. Here is your table, the genetic code. The first base is here on the left. So in this case here, you have the first base here, which is A. The top denotes the second base. So every base on the second row is going to be denoted by the base in that column. And then the third base is going to be the last one corresponding to this row. Okay, so in this case, it's U, U, and U. That's here. U, U, C is here. And I want to point out a couple of things. Notice in this box here, these two codons code for the amino acid phenylalanine. Whereas here, these 4 bases here code for serine. Okay, so there's more than one way to get the same amino acid. Okay, there's more than one way to get the same amino acid. The AUG here codes for the amino acid methionine, and again, it is the universal start codon. The universal stop codons are UAA, UAG, and UGA. And notice again, there is no corresponding amino acid. They only correspond to release factors to stop or terminate translation. As I mentioned earlier, the wobble referring to that third position that tends to be more laxed. And as a result of that, you can have instances where that third base does not always correspond to the codon in the mRNA, but you can still get the same amino acid. Okay, you can still get the same amino acid. So here are the wobble rules, and they help to indicate which base is in that third position. So the 3' end of the mRNA can code with which base is at that first position in the anticodon region. So you want to go back over this table 15.2 to just understand this a little bit better. So the last component that we talked about in that slide was the reading frame and the initiation codons. Okay, with the reading frame, there are 3 ways in which the sequence can be read in groups of 3. Each different way of reading encodes a different amino acid. So make sure when you're talking about a reading frame, it's referring to the 3 bases that are synonymous with each other, and they are read from left to right or in a non-overlapping manner. So if you have one nucleotide that's a part of one codon, it cannot overlap with another codon. It is read consistently without any pauses or commas. And this helps with understanding, when you look at it this way, how mutations can be incorporated because it is read consistently. So keep this in mind when we get to chapter 18 and start talking about mutations. The genetic code is also universal, and there's of course some exceptions, but for the most part, whether you're talking about a prokaryotic cell or eukaryotic cell, the genetic code is universal in how it translates the transition from mRNA to proteins, i.e. amino acids. Here are some of those exceptions to the universality of the genetic code. Okay. So let's go into now how amino acids are actually assembled into a protein through translation. The first thing you have is the binding of the amino acids to transfer RNAs. Okay, so the amino acids have to bind to a transfer RNA, and there's enzymes that help facilitate this process, known as amino acyl tRNA synthetase. That is the enzyme that charges or adds amino acids to the acceptor arm of the tRNA. And then, as we've seen with transcription and even replication, you can divide translation into 3 stages: initiation, elongation, and termination. So let's go through these. So translation of the mRNA is going to begin on the N-terminus. When you hear N-terminus, that's referring to the amino group, okay, the end, okay? And the N represents that amino end, and the C represents the carboxyl end. So here is your mRNA that's being used as a template to produce this growing polypeptide chain. This end is going to be the amino end. That means the amino acid that's added here, the amino group is on this end. And then the C terminus is going to be open on this end of the amino acid. And it is read in the mRNA is read in a 5' to 3' manner. As I stated, let me go back. The enzyme that charges the tRNA, meaning it adds a amino acid to the 3' end, is called amino acyl tRNA synthetase. Okay, and there is specificity between the amino acid that is being added and the tRNA. It's determined by that synthesis. There are 20 different enzymes, the amino acyl tRNA synthetases, that actually are responsible for charging these tRNAs. So here you see the tRNA here. There's your anticodon loop. There is your acceptor arm. And then you'll see here, there's the corresponding codons that are associated with the amino acid that will be added. Okay. Again, the carboxyl end of the amino acid attaches to the hydroxyl group of the 2nd or 3rd carbon atom of the final nucleotide at the 3' end of the tRNA, and in which this case is always adenine. So here you see the acceptor arm. Here's the anticodon arm. The positions in yellow are used by more than one synthetase. The positions in blue are the same in all tRNAs and cannot be used to differentiate one tRNA from the other. However, the positions in red are important because they help recognize the tRNAs by one synthetase. Okay, so these are the varying and the consistent parts of the tRNA. So the first step is the amino acid is reacting with ATP. Okay, so here's amino acid reacting with ATP and producing amino acyl AMP. Okay, so 2 of the phosphates are released. So it is an energy-driven reaction. Here's your tRNA. The next step is the amino acid is transferred to the tRNA. what you see here. And at the end of the charging, an amino acid is linked to its appropriate tRNA at that 3' acceptor arm. Again, it is the enzyme that is helping to carry that out, and it is a energy requiring or energy utilizing reaction. So let's go into now initiation. So now that we know how tRNAs are charged with carrying amino acids to bring them into the ribosomes, let's talk about how initiation of translation occurs. The first step in the initiation of translation is utilizing initiation factors. Okay, so in this case, IF3, and then you have the initiator tRNA that has the N-formylmethionine, or FMET is how you refer to it, and it also has an energy molecule, in this case GTP. Now let me be very clear and help differentiate the difference between one of the differences between prokaryotic cells and eukaryotic cells. In prokaryotic cells, the initiator tRNA has a formal group attached to methionine. And so whenever you're talking about a prokaryotic cell, you should always assume that if you see FMET or formal methionine, you are referring to a prokaryotic cell. Eukaryotic cells do not have formal methionine, a formal group attached to the methionine. Okay. The other component is the Shine-Dalgarno consensus sequence in the bacterial cells, and it recognizes that small unit. It recognizes that, and it's in the small unit of the ribosomes. We talked about this in chapter 14. In eukaryotic cells, it is called the Kozak sequence, and it helps to identify where the start codon is. So there are consensus sequences that help with the initiation of translation. It's the Shine-Dalgarno sequence in prokaryotic cells. It's the Kozak sequence in eukaryotic cells. So here you have the large subunit and the small subunit. This makes up the components of the actual 70S. Here you have the mRNA, and in the mRNA you have that Shine-Dalgarno sequence. You have the initiator factor or initiation factor IF3, which is bound to the small subunit, and it actually aligns with the Shine-Dalgarno sequence in the ribosomal RNA in the small subunit of the ribosomes. Once you have that bound there, you have the other initiation factors like IF1 and IF2 that come in with the initiator tRNA carrying formal methionine, and it binds to that start codon here. And once the initiator tRNA is bound and in the right place and is aligned with the mRNA, then you have all of the initiation factors that disassociate, and now the large subunit can come in and bind. A way to look at this is it's a compensatory mechanism. If the initiation factors are bound there, it's blocking the large subunit from being able to bind in that same place. So the initiation factors have to be released once the initiator tRNA has aligned in the right location to begin the initiation of translation. Okay, just to show you again really quickly, here is the ribosomal RNA that is embedded within the small subunit. This is the mRNA, and they align with each other through complementation. Okay, and this allows the small subunit to identify where to bind. All right, so here you have in a eukaryotic cell, okay, you have the start codon, you have the 5' untranslated region. Remember, we have the 5' cap that also plays a role in the initiation of translation, in part because it has the cap binding proteins that help with the initiation of that process. All right, so now we are going to transition into the elongation process, okay? Within the ribosome, there are 3 sites: the E site for exit, the P site for peptidol, and then the A site for aminoacyl. These 3 sites are strong indications of what's happening in each of these sites of the ribosome. Along with these sites, you have elongation factor 2, TS, and G that help with the process of elongation, bringing in these initiation factors and helping with the growing chain, primary chain of amino acids. Okay, let's go into the elongation step, and it's really broken down into 3 major steps. So first, you have here the initiation, the initiator tRNA that is bound to FMET, and it's going to always start in the P site, the peptidal site. Okay, translation always starts with a tRNA that is charged in the P site. You'll have an incoming tRNA that is charged with EF2 and GTP, and it will come in and it will be bound or bind into the A site. Okay, so now you have the P site that is full and the A site that is full. Once that initiator tRNA is in the P site and then the incoming charged tRNA is in the A site, then you have the EF2 factor, elongation factor, that is released. Then you can have the peptide bond that'll form between the two amino acids here, okay, in the P site and the A site. And then the tRNA that is in the P site will be released, and you'll have the translocation, the movement of the ribosomes down the mRNA to where now the tRNA that is carrying the growing chain is in the P site and the A site is left open. Something to keep in mind, and it's very essential. The A site has to always remain open. If the A site is ever blocked and you're not allowed to have the peptide bond to form and have translocation, it's going to stimulate termination of translation. Hence why you'll see in a second, when the machinery reaches a stop codon in the mRNA, it stimulates termination. So again, remember, the A site has to always remain open. So this brings us to termination. Okay, translation is initiated when the ribosome meets or comes in contact with a termination codon. That is UAG, UAA, or UGA. And the reason why this occurs is there's no associating amino acid. So another charged tRNA cannot come in with an amino acid bound to it to recognize either of these stop codons. And so in the place of a charged tRNA, in the A site, you will have a release factor that binds. The release factor does not have the ability to help with the process of elongation. Okay, so it stays in the A site and it blocks any incoming tRNA to come in and bind. And so because of that, it causes the release of that polypeptide from the last tRNA in the P site. The tRNA from the ribosome is released, and the mRNA is also released from the ribosome. Excuse me. So you can see here, translation ends when a stop codon is encountered. So here you have this growing chain.
Translate into which
You have this growing chain here. tRNA in the P site. It identifies UAG, which is a stop codon, and in that case, a release factor binds into that region. Once that release factor binds in that region, okay, you also have RF1 that'll bind to the A site, RF3 that'll be bound to GTP, and it helps to form that complex. Okay, it'll cause the dissociation of the complex to be released, and the tRNA and the mRNA will be released from the ribosomes. Now, with translation, okay, we talked about the tRNA charging, initiation, and termination. These amino acids are linked in the order that is specified in the mRNA. So it's important that the mRNA is read consistently. There are no mutations or errors in the mRNA, because whatever's in the mRNA will determine what polypeptide is produced from translation. Okay, so you see here that charging of the tRNA is essential for translation. Initiation factors are essential. The elongation process, you need a charged tRNA. And also what blocks translation, the elongation process, and stimulates termination is this stop codon. So all these factors are essential to the totality of the efficiency of translation. And any of these mutations and any of these factors can cause an abnormality or something abnormal to happen in the normal process of translation. Okay, so here you have that initiation process. There's your charged tRNA. There's your initiator tRNA bound there. Right, here's your elongation process. The EP, the E site is empty, and the E site is where an uncharged tRNA goes to before it exits. So that's the exit site. So once the amino acid has been transitioned or formed with the amino acid in the A site, then that amino acid is moved from the P site to the E site to exit out and be recycled. And then you have a growing chain that is now on the tRNA in the P site, and an incoming charged tRNA can bind to the A site. So these are just showing you the various steps of translation. I would use these last few slides that I'm showing you to really explain and write in your own words the process of translation and all of the components required for it. Okay, here is the elongation portion. Eventually, you have this growing chain of amino acids until it reaches a stop codon in the mRNA. And once it reaches a stop codon, again, you have a release factor that binds into that area, therefore causing termination or stimulating the disassemble of the translation machinery, thereby releasing the completed polypeptide, or in some cases incomplete polypeptide, and termination has ended. This slide here is essential. I will use this as a studying tool because it provides all of the components needed for protein synthesis in a bacterial cell. Know that these are going to be different for a eukaryotic cell. Okay. Just a couple of things that I want to talk about in addition to the process of translation. Okay. The three-dimensional structure of the ribosomes and the polyribosome, which is where you have an mRNA that has several ribosomes attached to it. So let's look at the actual structure of a ribosome. Here is your 50S, or your large subunit. Here's your small s. You'll see the mRNA, okay? And then you have the growing polypeptide chain. These are just showing you different models of how the ribosome structure looks. Notice here's the E site, the P site, and the A site. And keep in mind, you all, these are proteins that are involved in this, and this is a chemical reaction that's actually helping to anchor these molecules in place. Okay, so here you have an mRNA molecule that can be transcribed simultaneously by several ribosomes. So in A, you'll see here there are 4 ribosomes that are being translated or translating an mRNA molecule. The ribosomes are going to move from the 5' end to the 3' end of the mRNA. And here you're looking at an electron micrograph of a polyribosome. Okay, and in this case here, what you're seeing is the 5' end is towards the left and the 3' end is towards the right. But ultimately, this is showing a polyribosome, so an mRNA that has multiple ribosomes attached to it and carrying out the synthesis of protein. Now, there are some additional properties of the RNA and the ribosomes that affect protein synthesis. One, you have messenger RNA surveillance. And this is where you have the detection and the ability to deal with errors that may occur in mRNA. And this is essential because whatever is in the mRNA is going to be produced into protein. You also have what we call post-translational modifications. And this is where you have proteins that can fold in such a way or elicit the use of molecular chaperones to help with the proper folding. And this is essential because the protein has to fold into whatever shape it's supposed to be in its final shape so that it can be transported to its final location, whether it is in the nucleus, whether it is in the membrane, the docking in the membrane, or in the cytoplasm, so that it can carry out its function.